All right, welcome to chapter 3, which is all about the basics of genetics. So to start this off, we're going to just kind of give some background about genes, what they are, some vocab, and get into a little bit about different types of genes. So to start, we've got our basics. So DNA is the genetic blueprint of all organisms, right? So this includes physical, behavioral, and physiological features. Everything you would need to know about an organism you can find through their DNA. DNA is packaged into chromosomes, which allows it to fit into smaller areas. And a gene is a sequence of DNA that is specific on a chromosome, so a specific area on a chromosome that encodes for a specific trait, like eye color or hair color. And then, as mentioned, the position of a gene where it would lie on a particular chromosome, that's called a locus. Alleles, this is another big vocab word. So alleles are different forms of a gene. So same general idea of a gene, but different variations of that. So alternative forms of one gene, typically only different by one or a few base pairs. So an example of this would be eye color being the gene itself, and the alleles of an eye color would be like brown, blue, green, gray, hazel. Those are different variations of the same gene. Now mutations, there's a few types of mutations, and we'll get more into this later, but the general definition of a mutation is a change in the nucleotide sequence. So sometimes mutations can be beneficial, which just means that the change created new variations that are helpful to the organism in some way. It can be detrimental where they either truncate the gene sequence or they delete certain areas, add new areas, make the gene non-functional, and it changes from a functional to a non-functional gene, or a neutral mutation, meaning that nothing happened. So yeah, something changed, but the outcome was the exact same. Now, sickle cell anemia is an important disorder that we're going to study in more than one way. So sickle cell anemia causes an irregular shaped red blood cell, which has an effect on people's ability to fight off other diseases like malaria. But what this slide is really focusing on is the mutation that causes sickle cell anemia. And you're probably thinking to yourself, yeah, but like, you're telling me I need to know the exact mutation that causes this disorder? And I'm here to tell you, yeah, you do. You need to know the literal base pair mutation that changes, and you need to know what amino acid is normal and the sickle cell version of it. So you can see on this, the wild type, which is your normal hemoglobin, which is your blood, right? The ability to carry oxygen, is CTC. Its mRNA base pairs are GAG, and the amino acid you can see on the bottom. And then when you have this base substitution mutation, that T changes to A, which becomes U, and the amino acid also changes. So this is something you will be required to know. So let's talk more about sickle cell anemia. Like I mentioned, it changes the structure of a red blood cell, and it causes it to be unable to carry oxygen effectively. It also has a different shape, right? It's shaped like a sickle, which is why they call it sickle cell anemia. But because it's shaped so differently, it is its ability to form clots and block blood supply is pretty high because it doesn't flow smoothly through arteries and veins. So they also, because they aren't shaped normally, the body fights against them. So they're destroyed pretty rapidly from the body themselves. And so it leads to low red blood cell counts within the body, which is also a negative side effect of this. I mentioned a few slides ago that there's a relationship between sickle cell and malaria. And the relationship is if you have sickle cell, you also show increased resistance to malaria. So if you have normal blood cells, like you don't have sickle cell anemia, you don't have that any form of sickle cell, but you're more susceptible to malaria. But individuals with both alleles, right, you're heterozygous, so you have one sickle cell allele and one normal blood cell allele, you have enough normal blood cells to avoid the really bad side effects of sickle cell, and you have enough sickle cells that help show resistance to malaria. So it's like kind of making the best out of a bad situation. So you don't really have the full effects of sickle cell and you can fight off malaria. So there's a reason why this has kind of continued to persist in these regions shown at the bottom, because it's known as a heterozygous advantage. So it's advantageous to have an allele for sickle cell because malaria is kind of so rampant in these areas. So moving out of that and moving back into kind of just a vocab area, we're talking about genomes. So genomes is just all of your genetic information of a cell, an organism, or an organelle. It's just all of the bits that you need to know. So this is all genes, all non-coding DNA, everything that you can see in this picture on the right. It's like literally everything you would ever want to know. If we're talking humans, our genome contains 46 chromosomes, about 3 billion base pairs, and about 21,000 genes. Within those genes, there's also types of pseudogenes, which are kind of like fake genes. The two main categories we're going to talk about are transposons and pseudogenes themselves. So a transposon is a segment of DNA that can insert itself into another section of the genome. So basically it can take its segment of DNA and take it out of where it normally is and input it somewhere random. This can be both beneficial and obviously detrimental to organisms. A lot of fungi can do this, and there's a specific fungus that affects oak trees that the main way it can bypass the oak tree's defenses is by using transposons, by changing up its genome so that the oak tree does not, can't detect the fungus, and so the fungus goes in and wreaks havoc, and it's because they can manipulate their genome using transposons. Now, pseudogenes is just a non-functional sequence of DNA that's homologous to an active gene. There's two types of this. There's processed and non-processed. So processed is when the mRNA transcript is reverse transcribed back into DNA and then just randomly inserted. So you've just sort of made a backwards copy of DNA, and so it's non-functional because it doesn't code for anything anymore. And then non-processed is when you duplicate genes, and then when you do that duplication, you also have a mutation happen, which leads to inactivation. Usually these are located right next to the original gene because you've just duplicated that gene. Another big piece that you will need to know all about is the Human Genome Project. So this was an international cooperative venture to get the entire human genome sequenced. This is why we know what we know about the human genomes. This was completed in 2003, which is wild because I feel like this was huge news, and it was, but now it feels like a long time ago, but it still feels like one of those like big important things that happened in my life. I was a teenager during that time. So this Human Genome Project wasn't just for like the good of the scientists to be like, yeah, we did this. It also has so many outcomes that are hugely beneficial to the world. So yeah, you mapped the whole genome, so now you know like size and sequence and location of different things, but you can also screen new things. So now you can detect specific genes to determine if you are carrying a disease. You can add this to medicine so that you can improve treatments now that you know where these proteins are being made and where to target these treatments. And then ancestry, which you've probably seen rise in popularity in recent years. Well, now you can actually like figure out origins and evolution migratory patterns of like ancient ancestry, but also you can do this with people that are alive today. You can match your genomes because you're able to use the entire sequence that this venture made. So then you can kind of figure out, oh, okay, well, my family originated from wherever, and now we're in all of these different places in the world. It's pretty cool. And then last but not least is gene comparisons. This is just to kind of keep your head on straight. So the number of genes in an organism does not indicate biological complexity. Okay? So if you look at this chart, Homo sapiens, that's us, we're right in the middle with about 21,000 genes. And yeah, we've got more than chickens by about 4,000 genes. So like, yeah, okay, we have a little bit more genes, but we have way less than rice, okay? So it doesn't matter how many genes you have in an organism, it does not indicate biological complexity. That's something to keep in mind as we move forward through this unit.